Hi everybody, welcome back. So today we're going to be talking a little bit about action potential. We've discussed the idea of an action potential before and how it's this electrical charge that can move across the surface of a cell, but we haven't gotten into super detail about how exactly that works. So that's what we're going to cover in this video. Okay, so we remember that neurons are excitable cells, and that means that they can communicate using action potentials by sending an electrical signal along their surface and communicating that electrical signal to another cell to get it to react in some way. Okay, so I want you to think of your cells as being like a battery that have different ions on either side so that we've got a positive charge on one side of the cell and a negative charge on the other. So if we look at this graphic here, you can see your phospholipid bilayer, which is a really good insulator, right? It's made of fat, so that's going to protect ions from moving one way or another unless they go through specific ion channels. So in this graphic here, we can see that we've got some sodium ions, we've got some potassium ions, we've got some large proteins that are stuck on the inside of the cell. So those proteins, most of the proteins on the inside of your cell have a negative charge to them. So they're kind of holding a negative charge on the inside of your cell. And then we've got these sodium and potassium ions, which are both positively charged ions. So if we can think back to, oh boy, Unit 2 maybe, when we were talking about membrane transport, we talked about the idea of a sodium-potassium pump. So this is as a way of doing active transport where we're moving ions across the membrane using a protein pump. And the way that a sodium-potassium pump works is it's actually pumping in 2 potassium ions for every 3 sodium ions it pumps out. So 2 positive ions come in, 3 positive ions go out, and that's going to give us a net positive charge on the outside and a net negative charge on the inside, especially when we combine that with all those negative proteins that are sitting on the inside of the cell too. So overall, we get this resting membrane potential of negative on the inside, positive on the outside of the cell. All right, so if we actually measure that resting membrane potential, which we can do, if we actually measure it, it comes out to be about negative 70 millivolts. So negative 70 millivolts on the inside means we have a negative internal environment in that cell and a positive external environment. So this is where those ion channels are crucially important. And remember that sodium potassium pumps our active transport, that means that we need to use ATP in order to maintain this resting membrane potential, right? So we need to pay ATP in order to keep the balance of ions negative on the inside and positive on the outside, right? Because normally your cells would want to even that out, right? So we always want to even out on different sides of the membrane, whether we're talking about concentrations or charges. We always want to even out. So the fact that we're not even, the fact that we've got negative on the inside and positive on the outside means that we must be paying some energy in order to maintain that inequality. Okay, so hopefully we understand the idea of a concentration gradient. So again, remembering back to a few weeks ago when we talked about how diffusion works. So when you have a whole bunch of ions concentrated on one side, they're going to go from high concentration to low concentration. So we're going to move down that concentration gradient. So we also have an electrical gradient, which means that if we have a whole bunch of positives over here and a whole bunch of negatives over here, they're going to want to attract each other and kind of move down that electrical gradient as well. So we've got a couple different gradients working here. We've got the concentration gradient, but we've also got that electrical gradient. And if we put those two together, we refer to that as our electrochemical gradient. Okay. All right, so this maintenance of the resting membrane potential, or RMP, is going to be really important to the capacity of neurons to send messages, right? Because basically a resting membrane potential is just that. It's a potential source of energy, right? So it's just basically like rolling a whole bunch of balls up to the top of the hill. When those balls are at the top of the hill, they've got a lot of potential energy because all we have to do is let them go and then they'll roll back down. So think about your resting membrane potential as being just a whole bunch of ions that are pushed up to the top of a hill. right? They're holding a whole bunch of energy up at the top of that hill. And then as soon as we trigger them to release, they're going to roll down the hill, and that's going to be the message. That's going to be the action potential, is a whole bunch of ions rolling down the hill. All right, so think of your resting cell as being primed. It's in a state of readiness to send a message when necessary, because basically it's got a whole bunch of sodium ions on the outside that we're forcing them to stay outside of the cell until we tell them they can come in. Right, so they're beating down the door ready to get inside the cell because of the electrochemical gradient that wants them to come into the cell, but we're blocking them until the time is right. Okay, so when we're ready, so when a cell gets triggered, so a neuron gets triggered that something is happening, that's when we can possibly start the action potential. So usually what happens is, imagine your neurons. Let's talk about a neuron that feels touch in your skin, for example. So when I touch my skin, what that's going to do is disturb the neurons there. It's going to disturb the neuron to the point where it's going to start opening up some ion channels. So when the neuron gets triggered, it's going to open these ion channels. When that happens, remember, we've got all of these ions that are kind of just waiting on the outside of the cell to be let in. So once we open the ion channels, sodium ions are going to start rushing in. So if we get a few sodium ions coming in, that's going to make our resting membrane potential go up a little bit. So I want you to look at this diagram here. So normally we're at -70, right? That's our resting membrane potential. If we trigger the neuron by touching it, or if it's, say, a neuron that's in your eye, we can trigger it by light hitting it. If it's one in your ear, we can trigger it through the mechanical motion of the sound. We can trigger different neurons in different ways, but as we trigger it, we're going to open some of those ion channels, and as we open them, some sodium ions come in, and we get a little bit less negative on the inside of the cell. So we might go from -70 to -65, maybe to -60, right? So as we trigger that cell, more and more sodium ion channels are going to open, letting sodium rush into the cell. If we get enough sodium coming in that we hit this -55, right? So -55 we consider our threshold. So after we hit -55, it's going to trigger all the ion gates to all those ion channels to open wide. And that's going to cause a huge flood of sodium ions inside the cell. So we can see after we hit that threshold, then we get way, way positive, right? So that we actually get so many sodium ions flooding into the cell that now the inside of the cell is even more positive than the outside. So we get all the way from -55 up to +30, right? So we've got a whole bunch of positive charges on the inside of the cell now. That's our action potential. So our action potential is when we reversed the polarity of the cell. It went from being negative on the inside, now it's actually more positive on the inside than it is on the outside. Right, so we get this huge reversal in charges. That's the action potential. That's a signal, an electrical signal that's going to travel along the surface of that neuron. Right, now, after we've done that, after we've triggered that action potential to happen, now we have to get back to resting. Right, so we've triggered that neuron. Now we have to get it back to normal so that it can be triggered again the next time I go to touch something or see something or hear something. Right, so in order to get it back to normal, we got to gear up those sodium potassium pumps again and start pumping a whole bunch of sodium ions outside of the cell so that we can go back to normal. So that process is the repolarization, where we're kind of going back down the hill, right? So we're pumping a whole bunch of sodium ions out. Sometimes we overdo it and we go into hyperpolarization where we're even less than -70, but then it usually corrects back out and we're back to resting membrane potential of 70 millivolts, or I'm sorry, -70 millivolts. Right, so here's a closer look at that repolarization phase. So as soon as we've kind of hit that maximum reversal of potential, then we have to get back to normal by pumping a whole bunch of sodium outside the cell. Also, potassium is going to rush outside the cell as we try to reestablish that electrochemical gradient. So we want to get back to negative 70 millivolts. And until we do, the cell's in a refractory period. So that refractory period is a period where we cannot re-stimulate that cell. We can't re-initiate an action potential until we've gone back to normal, right? So we have to repolarize, get back to normal. And while we're doing that, the cell is in a refractory period. Okay. We've talked a little bit about what happens at synapses before. So we talked about what happens at a neuromuscular junction, which is a type of synapse. And essentially, a synapse is just a way of carrying the signal that was electrical, right? So we had an action potential that was moving along the surface of the cell, and that hits the end of the cell. When it hits the end of the cell, then we have to convert that electrical signal into a chemical signal, right? So what happens when we hit the end of the cell is we trigger a whole bunch of calcium channels to open. Calcium ions are going to rush into the cell, and that's going to trigger the release of the neurotransmitter. So whatever neurotransmitter we have in those synaptic vesicles at the end of the axon terminal, that's going to get released into the synapse. Those neurotransmitters then take that chemical message across the synapse and can possibly reinitiate an action potential on the postsynaptic neuron. All right, so here's what that looks like. So we've got our action potential moving down the axon here. As we get to the axon terminal here, we're going to trigger a whole bunch of calcium ion channels to open. So that means all these calcium ions are going to be flooding into the axon terminal. That's going to trigger these synaptic vesicles to undergo exocytosis. So those synaptic vesicles are going to merge with the cell membrane, push the neurotransmitter out into the synapse. Then those neurotransmitter molecules are going to migrate across the synapse until they hit receptors on the postsynaptic neuron, so the next neuron that comes after the synapse. Ideally, then those receptors are going to trigger a new action potential to begin, right? So it's kind of, this is one neuron disturbing another neuron enough to cause ion channels to open so that we can get a new action potential to happen in that postsynaptic neuron, and that's going to carry the message through that next cell. Okay, so just a quick note on neurotransmitters. There are a few different types of them, right? So we've got neurotransmitters. We've talked about acetylcholine before and how acetylcholine is the main neurotransmitter of your somatic nervous system to get muscles to contract. But we've got a whole bunch of neurotransmitters. Some of them are excitatory, which means they perpetuate a signal, send that signal along. Some of them are referred to as inhibitory, and inhibitory neurotransmitters are ones that are going to stop a signal. So there's signalers that say, stop, don't go forward, right? And sometimes neurotransmitters can behave different ways depending on which synapses we're talking about. So acetylcholine, for example, is excitatory for your muscles, but it's also inhibitory at some other places, which we'll talk about later on. Okay, so neurotransmitters can be they can be modified in different ways. So we can create more neurotransmitters. We can release more neurotransmitters. Sometimes we can stop the release of neurotransmitters. Sometimes we can stop the uptake of neurotransmitters by the postsynaptic neuron, right? So, and we've got lots of chemicals that can kind of moderate the effect of these neurotransmitters. So an agonist is a chemical that's going to enhance the effect of that neurotransmitter. An antagonist is going to block it. So some are going to be agonistic, which means they kind of work together, and some are going to be antagonistic, which means they block one another. Okay, so a lot of these neurotransmitters are small molecules called amines. Amines contain ammonia. They have a nitrogen component to them. So some of these amine neurotransmitters are going to be important for your psychological disposition. And you've probably heard of MAOIs before, or monoamine oxidase inhibitors. So these are a type of antidepressant drug that stops the breakdown of neurotransmitters in the synapse. So a lot of times these neurotransmitters get, as they float across the synapse and they reach the postsynaptic cell, sometimes we have a whole bunch of those neurotransmitters that are just floating around in the synapse, and you have enzymes in there that help break them apart to get rid of them, so we can kind of clean up the synapse a little bit. So a monoamine oxidase inhibitor is one that's going to increase the amount of that neurotransmitter in the synapse because it stops it stops the breakdown of that neurotransmitter. So it stops the enzyme that would normally be breaking it down and dissolving it. Similar thing is true for SSRIs, or selective serotonin reuptake inhibitors. So this is for the neurotransmitter serotonin. So again, it's going to increase the amount of serotonin that you have in the synapse by stopping the reuptake. So normally your cells would try to get that serotonin back inside instead of leaving it in the synapse. But an SSRI is going to leave a little bit more serotonin in the synapse, which hopefully boosts your mood a little bit. Okay, so a few questions here. When we're depolarizing, what is going to open during the depolarization phase of the action potential? Question 2, what flows into the nerve cell during the depolarization phase? What phase follows the depolarization phase? And then what's actually happening in the phase that follows the depolarization phase? All right, so here's your answer. So the thing that's going to open in depolarization is ion channels. Specifically, a whole bunch of sodium ion channels are going to open. And so that's going to allow sodium ions to flow into the cell. So that's what flows into the cell during depolarization. After we're done with the depolarization, and now we're all the way up to that positive 30 millivolts, then we have to do the repolarization phase, where we get back down into our negative resting membrane potential. So repolarization follows depolarization. And then what's actually happening in repolarization? Well, we're pumping a whole lot of sodium back outside the cell in order to get that negative resting membrane potential. Okay, before being released, where do we find neurotransmitters? Where are they stored? Yep, they're in vesicles in the axon. So we find little vesicles that are storing these neurotransmitters in the terminal bulbs of the axon. All right, so let's talk a little bit about sensation. Now that we kind of get the idea of an action potential, let's talk about how we feel things. So just as a general overview for how the nervous system works, we've mentioned this before. Remember that you've got sensory neurons, and sensory neurons are afferent pathways that are carrying messages to your central nervous system. And then we've got motor neurons, or efferent neurons, that are carrying messages away from your central nervous system. And then in between the afferent and the efferent neuron, we have an integrating neuron that's going to take that information, decide what to do with it, and then send out the message that follows. So in this pathway here, let's look at some, these look like Merkel cells that are in the dermis of your skin, right? Those are going to be able to feel touch. So if I apply some pressure here to the skin, so I push down on the skin, it's going to trigger those Merkel cells. That's going to go up an afferent pathway. So it's going to travel up this afferent pathway. We go into a nerve. We're going to go all the way into the central nervous system. So here's a cross section of the spinal cord. So into the nervous system, that's going to carry us up to the brain. Pretend your brain's up here. Your brain's going to integrate that information and then send a message back out. It's going to go all the way back out through that same pathway, but now this is an efferent pathway to some muscle here. So this muscle is going to be reacting to the touch. So if I touched myself with something sharp, that message is going to trigger that afferent pathway. The afferent pathway is going to go all the way up to my brain. My brain's going to decide, ouch, that hurt. It's going to send a message down the efferent pathway and to say, okay, flex these muscles to pull my hand away from whatever it is that just hurt me, right? So it's a kind of a circular pathway where we're starting at the afferent end of this, we're sending the information to the integrating neuron, and then we're going back out to an effector cell. All right, so a little bit of information on the difference between somatic senses and visceral senses. So somatic senses are the things that you normally think of when you think of sensation, right? So touch, pressure, vibration, itchiness, tickles, the feelings of warmth and cold, feelings of pain, those are all somatic senses. And in addition to those, there's another sense here called proprioception. So proprioception you may not have heard of. These are tiny sensors that allow you to feel where your muscles, where your limbs, where your joints are in space. So the fact that if I raise my hand, I kind of have an awareness that my hand is raised, and that's because of proprioceptors that we find in the muscles that are associated with that movement. Right. So those are all somatic senses. And we also have visceral senses. So when you think of visceral, think viscera kind of like your internal organs. So visceral senses are letting you sense things that are going on inside your body. All right. And again, the sensory pathways are going to be similar for these, but you may not be able to have as much of an effect on your internal visceral reception as you would on the somatic receptors. So again, here's your pathway. We start with some sort of sensation, and right here they're showing you touch on the skin, but this could easily be vision, it could be taste, it could be smell, it could be sound, could be a lot of things. So we send an afferent neuron here is going to take that information, and then we're going to hit a synapse. At the end of this afferent neuron, here's our synapse between the afferent neuron and the interneuron. So once the interneuron picks up that information, it's going to pick it up through its dendrites, send it across the cell body all the way down its own axon, and then it's going to go to this next synapse. So here's a synapse here that's between the interneuron and the efferent neuron. The efferent neuron will then pick up the signal that it's getting from the interneuron. It's going to go through its dendrites, cell body, and axon all the way out to an effector cell. And at that effector cell, then we have another synapse there, right? So that's the neuromuscular junction that we talked about last week. So the synapse between the efferent neuron and the muscle cell that's going to trigger that muscle cell to do some sort of action. Okay. All right. So different sensory receptors, we've got a lot of them, and they can feel a lot of different things, obviously. So some of them we classify based on structural differences in the actual the structure of the receptors themselves. So some nerve endings we consider free nerve endings. Some are encapsulated. So encapsulated nerve endings would be like your Merkel cells. So those Merkel cells that are in your dermis, they have kind of like a covering around them. They're encapsulated. Whereas some of the receptors that feel pain, those are free nerve endings. They don't have a capsule around them. Sometimes we refer to these receptors by their location. So whether they're exteroceptors would be on the external surface of your body versus interoceptors or visceroceptors, which are on the inside of your body. And then sometimes we just classify them by the type of stimulus that they detect. So for example, nociceptors detect pain, mechanoreceptors detect pressure, etc. All right, so yeah, exteroreceptors are ones that are located near the external surface of the body so they can respond to external stimuli. So stimuli from the environment coming into your body are picked up by exteroceptors. So if I feel cold, it's because exteroceptors are telling me that the temperature around me is dropping. Interoreceptors, also called visceroreceptors, those are on the inside of your body. So those are going to send messages to your brain to tell you things like what's your blood pressure right now, what's your internal temperature, what are your muscles doing then in terms of digesting and moving food through. So that kind of internal information that your body needs to know. And then proprioceptors, which again I mentioned, those are in your muscles and tendons. They're going to provide information to your brain about where your body is in space. So just kind of locating where your body is in space and knowing that, hey, I'm sitting upright and not sitting upside down, for example. Okay, and then so just a whole bunch of different type of sensory receptors that we can classify based on the type of stimulus that's going to trigger an action potential. So mechanical receptors here, that means that we're going to have to either put pressure on them, touch them, we're going to move them. Some sort of sound wave is going to trigger them, a vibration will trigger them, or even just gravity will trigger them. All of those are mechanical sensations that will trigger the beginning of an action potential, and then that action potential will be sent to the brain to interpret. For your thermal receptors, you've actually got different thermal receptors that can feel heat and different ones that feel cold. So it's not the same cells that feel both of those different things. It's different cells feel heat, different cells feel cold, and you've even got some that can detect infrared radiation. Chemical sensors, these are things that you would find in your nose and on your tongue that allow you to taste and smell different chemicals. And then you've even got some electromagnetic receptors that allow you to feel light. Well, just I should say to sense light, so that you can sense that's your sense of vision, and even electricity. All right. So a lot of these receptors are named after the thing that activates them. So I mentioned mechanoreceptors. So you have to do some sort of mechanical action to change the shape of a mechanical receptor, a mechanoreceptor, before it'll trigger an action potential. Same thing with a thermoreceptor. It needs to change temperature. Nociceptors are for pain, so that they respond to damage is what they respond to. So any kind of tissue damage is going to be picked up by a nociceptor. Photoreceptors are activated by light, chemoreceptors are activated by chemicals, and then osmoreceptors detect osmotic pressure in body fluids. So those are things that are going to be able to detect like things that are in your blood pressure, and so on. All right, so let's talk a little bit about nociception. So nociceptors, again, are responding to damage. So anytime there's been damage, a nociceptor is going to send that message to your brain that you're in pain, and that allows you to, it's adaptive, it allows you to move away from the thing that's hurting you or causing damage to your body. Okay, so two different main types of pain are fast pain and slow pain. So fast pain tends to be really well localized. So this, a lot of times people refer to it as like a sharp pain. You know exactly where that fast pain is happening. Slow pain is slower. People usually describe slow pain as being more of a dull, aching, throbbing sort of pain that seems to last for a longer amount of time. All right, so that's the difference between fast and slow pain. Okay, and then the difference between superficial somatic pain and deep somatic pain. So superficial somatic pain would be, you know, if I have a cut on the surface of my skin, right? So something like that's going to be really superficial. Deep somatic pain is going to be when you go down below below the integument and go into muscles, go into the bones, joints, tendons, all of that's going to be a deeper kind of somatic pain. And then anything below that, so when we get into the viscera itself, that's where we get into visceral pain. So visceral pain a lot of times can be hard to pinpoint, right? So when something is painful inside our viscera, it can be hard to tell where exactly that pain is coming from. And so a lot of times your brain will interpret the pain as being somewhere that's different from where the damage is actually happening. So that is the idea of referred pain. All right, so let's look at some referred pain pathways here. So the one that probably stands out to you is the heart there. So and this one is pretty commonly known, right? If you're having a heart attack, any kind of damage to your heart, you usually feel the pain in your left chest, but then also down the left arm, right? So we can feel that pain not just where the actual heart is, which, you know, the actual heart would be right around here. So not just where the heart is, but kind of a more superficial and over to the left-hand side of the body. So heart pain tends to be referred on the left chest and left arm. Similarly, pain in the lungs or the diaphragm a lot of times is referred to the left side of your neck and left shoulder. You would feel pain there. For the liver and the gallbladder, it's going to be over on the right neck and right shoulder. A lot of times that's where the referred pain from those organs come from. If we go down a little bit further, we can see Bladder pain we usually find in the pelvis, but also on the inner thighs, right? Your bladder is not in your thighs, but sometimes the pain is referred there. Um, Pain in the ovaries tends to be referred a little bit higher than where those ovaries actually sit in the body. So referred pain is just your brain is interpreting the pain as coming from a different place than where the damage is actually coming from. Okay. And then let's talk about proprioception just real quick here. So proprioceptors again are these muscle spindles that you have in your skeletal muscles that are able to tell where your muscles are in space, and they basically do this by measuring stretch. So if my muscles are if I'm doing this action, for example, so I'm flexing my elbow, that means the tendons that are attached to my triceps brachii are stretched around the surface of that joint right now. So that's how my brain is going to understand that I'm stretching that my arm is moving in that way, is because those stretch receptors that are in the muscles. Okay. A quick note on phantom limb. So this is one sometimes students have questions about. When a patient gets a limb amputated, sometimes they can still feel that limb, so they feel the pain in that, and that's because there's still going to be nerve endings that would have gone to that particular limb that were then severed when the limb was amputated. And your brain doesn't know what to do with that information. So sometimes it still kind of interprets the limb as being there because there's still pathways toward it. It's just that they don't go all the way to where they would have normally ended. So it's still stimulating your brain, and your brain's trying to make sense of the fact that those pathways no longer go where they used to. All right, so a couple questions here. So of these different senses, which are going to be somatic senses and which are visceral senses? So proprioception, organ stretch, pain, blood pressure, temperature, and then tactile sensations, which means touch. All right, so proprioception is a somatic sense, so that's the feeling of your body movement in space. Organ stretch is visceral. So organ stretch would be like if you ate a really big meal and you can kind of feel like your stomach's a little bit stretched out. That's a visceral sense. Pain is definitely a somatic sense. Blood pressure is visceral. So the feeling of the blood pushing more strongly on the inside of your vessels. That's a visceral sense. Temperature is somatic, and tactile sensations are also somatic. Okay, what are nociceptors and what do they detect? What is referred pain? And then where do you feel the referred pain for your lung and diaphragm? Where do you feel it for your heart? Where do you feel it for your liver and gallbladder? And where do you feel it for your urinary bladder? All right, so nociceptors are pain receptors. They detect damage. So they don't detect pain. You feel pain, but pain is your way of interpreting damage, is your brain's way of telling you that there's been damage somewhere. So referred pain is where the damage is happening in one place, but you're feeling the pain in another place. And then where do we feel the referred pain for these different places? Lung and diaphragm, you're going to feel pain in the left neck and shoulder. Your heart is going to be the left chest and left arm. Liver and gallbladder is probably going to be over on the right side, right neck and shoulder. And then urinary bladder pain is going to be in the pelvis and inner thigh. All right, so I hope this was helpful. Let me know if you have any questions.